So we're going to continue talking about transcriptomics today, really landing on the main method that is starting to be predominantly used at this point, which is, and is expanding all the time, which is RNA-Seq, which we slightly got into on Monday, but we really didn't get to go into this. You might think I don't need this anymore because I keep starting it before I hit this slide, but in truth what happens is I go to look at this, and I have my little queue of slides, and I see it up, and that reminds me. So I still use it. Okay, so RNA-Seq right now is most commonly used, or I'm going to claim at this moment from some brief searches, and I'll give you some evidence, that seems to be the most commonly used transcriptomic approach currently used. Is also the newest and one of the most powerful, which kind of makes sense. So just to give you an idea of how much is being used, one of the things that really impressed upon me that this is a really important transcriptomics approach was this last January when I went to the SICB conference, which is the Society for Integrative and Comparative Biology. It's a fun, big conference with lots of people. And it tends to be lots of people studying other organisms other than humans, like not people, generally not dogs and cats either. I don't know why I thought some dogs and cats. Maybe some dogs and cats, but a lot of kind of like basic biology on non-model systems, and yeah, it's doing a lot of cool stuff. But just to kind of give you an idea. So what's cool about that is that there's like a whole range of approaches that are being used. There's everything from ecology and like ecosystem studies to very like transcriptomic studies that are being done all the time, which is really interesting. So I enjoy that. And then it's just being done on all these cool animals. I don't know if any of you know who Kevin Padian is, but he gave a talk on Quetzalcoatlus, an extinct pterosaur that was really huge, and he gave a talk on how it walked on land. So it's like some biomechanics stuff. Anyways, cool stuff. Anyways, so in SICB, I just because I had the abstracts, I decided to go through and tally up how many talks or posters were using each of these transcriptomics approaches, and if you look, RNA-Seq was used by 39 talks or posters. There's not a huge amount. It's not like everyone there was doing transcriptomics, but there's a pretty good representation, including someone that I went to graduate school with I bumped into there, which is kind of cool, and he actually won the Best Student Award. He was using RNA-Seq to look at differential expression of genes, and so he's looking at beetles and like beetles with really big ornamentation, like huge horns and stuff like this, and he's looking at both a lot of different things, but one of the things that he did is he used transcriptomics to look at some of them have a morphology where they have a really big horn and some have smaller horns, and looking at the transcriptomics of how that happens. Anyways, so a lot of them were using RNA-Seq. Some of them were using microarrays, but like far fewer, only 4. And then express sequence tags were being used by one poster. Actually made reference to express sequence tags. Again, this is just in the abstract, so there could have been more one way or the other actually in the actual talk of the paper, but this is just from the abstracts. So, okay, that's interesting, but this is kind of like one subdivision of biology. Sure. So I also went and looked at a journal that tends to publish things like this a lot, BMC Genomics. And the skew isn't quite as big there, but in 2016, so up to date this year, in the abstracts of the papers, in the abstracts of the papers they published so far, 182 of them have mentioned RNA-Seq, 152 have used microarrays. So there's still, it's about on par, but RNA-Seq is still kind of outstripping it there. And express sequence tags were used in 26 papers. So just kind of reiterating, kind of just giving you an idea of the relative contribution of each of these techniques two current transcriptomics. RNA-Seq right now is being used more often, it seems, in major venues than microarrays or ESTs, or really both of them put together. So just a kind of recap of what I talked about on Monday. RNA-Seq is using high throughput RNA sequencing. Generally, this is Illumina sequencing, and so I'm going to briefly go back over that. Because this is important. How Illumina sequencing works is important to actually how we interpret this data. Because Illumina sequencing does some unique things that makes RNA-Seq data somewhat unique to analyze. So we gotta kind of refresh you on that for just a moment. So just to remind you, I want you to go really quickly go back over the Illumina sequencing process. It's also, this is the reversible dye terminator sequencing, and again, what we are doing is we're taking DNA, whatever DNA we want to. Now, in this instance, we're going to be using the RNA, and I'll talk a bit about how we get just the RNA or the messenger RNA that we want. And then generally we amplify it, and then we're going to chop it up by transposomes. So and this is called tagmentation, so we're actually fragmenting the DNA that we do have into smaller chunks. And then adding an adapter to both ends. So we're ligating then on a known sequence to both the beginning, the 3' and the 5' end of all of our DNA. Then washing it over a flow cell that has a ln in this particular lane. This ln is going to be of all these oligonucleotides on there. And we can then create colonies by essentially using PCR again. So I'll show you a picture again of how this works. And the nucleotides that we have added now have these reversible inhibitors, so that we add one single nucleotide, and then we can, it will stop with just that single nucleotide because it has this reversible inhibitor bound to it. We then also have a fluorescent tag on that, so we can fluorescently see which nucleotide was added. We can remove the inhibitor, adding from that one attached nucleotide, and then add another one, so we can step by step add additional nucleotides to our chain and fluorescently see what is the new nucleotide being added. So again, just kind of to remind you, we have a flow cell. This is looking on it end on, and we have all these oligonucleotides, and generally we have two different kinds there. Each of them are going to then bind to one of the adapters. And then, so here's our DNA. We have the adapters on each end. It's going to hybridize with one of the oligonucleotides in our flow cell. And then what happens then is then it will bend over, and we can get to hybridize then with another oligonucleotide that has the opposite adapter on it. And so what we get then is we have all of these bridges. Stick a primer on there, and then we can actually go through and duplicate this so that we can run the entire sequencing. So, well, back up. We duplicate this, and so actually now we have two. And we keep running this over and over and over again until we have an entire colony of exact matches or exact copies of that original piece that hybridized there. So what you get on the flow cell then is a small area or a small spot that has all been populated by the exact matches of what originally was there. Now, all that being said, once you get this, you're going to chop off half of these ones with certain ones. So we only have one direction on here, and then we go ahead and we sequence this using that technique that we're using, where the synthesis by, or sequencing by synthesis. We add one, look at the fluorescence, add another, look at the fluorescence. Now, here's where this is a little bit more extended than what I've talked to you about before. Once we sequence one direction, so we can get the entire sequences, what we can do then is now hybridize this again with the other oligonucleotides on the flow cell. So we got that, this up again, and we can get the complement side of this, chop away the original again, and then we can sequence the other direction. So one thing that you probably noticed when we sequenced your stuff is that the farther it goes along, especially if it's a really long one, it gets worse. Like, your ability to actually confidently call bases after a certain amount of time just gets bad. And that really happens basically with any kind of sequencing. You have good reads at the beginning, and then as it gets longer and longer, the quality of the reads goes down. Same thing happens here. As we're sequencing, we get good reads, but then further and further down, we start to get messier and messier reads. So what we can do then is essentially, again, hybridize this, do the complement strand, essentially go back to this, chop off all of the other side strands, and now what we're doing is we are now sequencing the other direction. So we come back to here, and now we are sequencing going the other direction. So what we can get is for every one, each one of these clonal spots, we can sequence one direction, get our sequences out, and then we can essentially turn the DNA around. Actually, it's not turning the DNA around, it's making the complement strand. And then sequence the other direction again, and then we have essentially going from both directions the same DNA, or the same sequences. So that is very useful. We'll talk about that more in a little bit. So the great thing about Illumina is that we have really high throughput. Now this is essential for RNA-Seq because when we're looking at tons and tons of transcripts, we need to be able to sequence lots and lots and lots of chunks of DNA. And the other thing that makes this really useful for RNA-Seq is that we only need one copy of each target to then hybridize to that flow cell, and then we can amplify it essentially on the flow cell. So this is, again, what makes it really nice for RNA-Seq is that we don't have to have tons of copies of it. We can actually have very, very few copies of each transcript, and we still can get sequencing on that. And it's easily automatable. Now the other thing is that we don't get very long read lengths with this, only somewhere 50 to 300 base pairs. We cannot sequence with one go an entire transcript. An entire messenger RNA is going to be hundreds to thousands to sometimes tens of thousands of bases long. We cannot sequence that entire transcript. Hence why we have to chop that thing up and look at it that way, but we can get little chunks of it. And lots, I mean, lots of little chunks of it. And then the accuracy is really high, which is going to come back, and we'll talk about some comparable methods later. And it's relatively cheap per megabase. That being said, if you're doing an entire transcriptome, this can get somewhat expensive, especially if you're having those people do all the prep. Okay. What you get out of this then is a fast Q file. What you would get back if you were to send something in for RNA-Seq, and then what you get back is a fast Q file. Now, this is a file that I talked about earlier on, way back when we were talking about different file formats for DNA. And we've used several of them. We've used Nexus, we've used FASTA files, we've used other things like that up until this point. So don't really need to refresh you on those, but this one I talked about and we haven't mentioned since. So I want to just refresh you on the FASTA file, especially since we're going to be using them in the lab this week. So this is similar to FASTA, but what we have that's different than a FASTA file is that we have quality data now that's been added to the file. So instead of beginning with is greater than sign that begins the header line, we have an @ sign that begins the header line. Then after that, you have one line for the genetic data. You then have a plus, and then you have one line for the quality data. And then you have a whole bunch of different characters that are just ASCII characters that represent all of the different qualities that you can have. And so it looks just it looks like more jumbled than regular expressions. There's just random characters up there. It looks like one long curse. And this is what it looks like. So this is an example of actual RNA-Seq data in a FASTQ file. And again, up here you have the @ symbol. So this is your header. This is the actual kind of stuff. This is some stuff about the run, is Illumina sequencing, and then you have some more tags that tell you exactly what spot it was and things like this. Then the next line, you have this one line that actually gives you the actual sequence data. And again, you'll notice this is short. It's a lot shorter than what you got back for your Singer sequencing stuff that we had done for your goodness. And a plus, and then you have a line of just what looks like random characters. Each of these random, semi-random characters, plus to some specific quality that we can we can map to, oh, how good is this read, right? This one specific base call. And then there's no space, and then it just goes right into the next one. So this is one set of data. This is a second set of data right here. This is your third. This is your fourth. So there's 4 different sequences through here. I think this one particular file had something like 15,000 lines on it, and it was a shortened one. So that's something in the neighborhood of like 4,500 different transcripts in there. Okay, so kind of getting back now then to the actual RNA-Seq. So what we're using here is this high throughput RNA sequencing, and generally it's Illumina. And what this gives us then is either paired or single reads. We can go through and actually get a single read, which is one direction that we're talking about with Illumina, or we could have paired reads, which is that we have both ends going towards each other. And just like we have in Illumina, we have our mRNAs that we're going to isolate, and we're going to isolate those through 2 different methods. We're going to have poly-A enrichment, which is essentially tagging or using some method, and there's quite a few different methods of doing this, of attaching on to the poly-A tail of the mRNAs, and then washing everything else out. So some have them that they actually have a poly-T section on to hybridize to, then that's attached to a magnetic bead, and actually use like a little magnet to like pull out the mRNA transcripts a few different ways. But this is really good and really specific to mRNA. Now there's advantages and disadvantages to each of these methods I'm going to talk about, and they give you slightly different data, so you have to be aware of that. Poly enrichment will give you is really specific to mRNAs. However, that being said, it does not necessarily get all of your mRNAs. Like I talked about on Monday, you have some mRNAs that do not have poly-A tails, and so it will not grab those. Now those are in the extreme minority, but you have to be cognizant if you're running this way that you are not getting all of your mRNAs. The other direction, because of that reason, the other direction that people have gone is just to break down or eliminate all of the ribosomal RNA, called ribosomal degradation, and then we take all of the RNA that is not ribosomal RNA. Now, the part about this then is that you're getting more than just your mRNAs. You also have microRNAs in there. You also have like long non-coding RNAs. You have all these other RNAs that are in there as well. Now, that being said, you can use filters after sequencing to get rid of some of those or a lot of those, and you can still get down to just your messenger RNAs by and large. However, that being said, that's more computation time. So each of these has hurdles that you have to get over after this, but that being said, they both work pretty well. And the more common of these seems to be the poly-A enrichment. Just in my brief kind of saunter around RNA-Seq literature. And then what we get then is hundreds of millions of short reads that are kind of in the range of what you would expect from Illumina sequencing, somewhere between 50 to 200 or 50 to 300 base pairs long. So you got millions upon millions upon millions. I guess upon millions means times or plus, I don't know. When you hear millions upon millions, do you think a million plus a million plus a million, or do you think a million times a million times a million? Plus? That's what I think too, but then I just thought, okay. So millions upon millions upon millions. So it's lots of millions of all these short reads, and then you're like, what do I do with these? Now, what we then need is to be able to then reconstruct these transcripts or reconstruct these reads into the actual transcripts. We need to somehow be able to make this all. How's it going? Sleeping, yeah. Anyway, so what we need to do is we need to be able to take all these short little reads and turn them or reconstruct them into the transcripts that they came from. And there's a couple ways of doing this. One of the main ones is just to map this then to a reference genome. So we can actually take all of these, if we have the genome, if we have a well-annotated genome, which is even better, we can take them and we can just align all these transcripts to the genome somehow and then say, oh, hey, this is that. quick and easy. However, that being said, if we don't have a reference genome, we can actually take overlaps between these and start reconstructing them back just de novo into the transcripts as well. A lot more computationally expensive, a lot higher error rate, but that being said, you don't necessarily need a reference genome to reconstruct your data into the transcriptome. So I'm going to really quickly talk about paired versus single end reads. So what you have then is with Illumina data, you can get these paired end reads where we're reading the segment or that tagmented stretch of DNA from two different directions. And this is nice because if you have paired ends, you have three essential different sets of data. You have the read from the 3' to the 5' end, so to speak. We have from 3' to 5'. Now, that being said, you're actually still sequencing the same direction, but because we're doing the reverse complement, you have that. And then we also have the whole read length, or the entire chunk of DNA that you're looking at, you have that whole length as well, which is also really nice. And this can improve, number one, it can improve the length of your read, because if you just go one direction, you're going to start getting unusable data at some point. You go from the other direction, you can get unusable data from going at some point, and hopefully you can get some additional stuff on there. That being said, this also improves your alignment. So one of the things that can happen is if you have a stretch of DNA that has AT, 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 and you don't know how long this AT goes on for, you can have a lot of reads that will be something and then end in AT, 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 and then a whole bunch that start off AT, 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 goes off. And it can be really hard, especially if this is very long, to be able to tell exactly how long this is, because you don't get any reads that span the entire thing. You have some that end in there, you have some that start in there, but you don't have any that actually span the entire thing. So if we have something that's like a tandem repeat here that's like 150 base pairs or 100 base pairs, it can be fairly common for you not to get anything that spans the entire thing, so you have no idea how long the actual thing is. If we have our paired end reads, this helps alignment in these sections significantly, because A, it's going to lengthen our reads, and then also, even if you don't get a good total length of a read, so for instance, you have good reads here, all of this was unusable, but you know there's base pairs here. We have another read going this way, and then we have this unusable, but we know there's base pairs here. We have a total length, and we know it's here, and we know it's here, and we know the total length of this. Now all of a sudden we can start getting a good estimate of how long these things are, despite not having any good single read that's going through the entire stretch of tandem repeats. Does that make sense? So that can work? No. What doesn't make sense about it?
I mean, I zoned out for like 5 seconds, so I might have missed the crucial part.
Okay, that's fine. I will go through it again.
It's all good, it's all good.
So, long stretches, hard to actually get things that span if they're very long, like a few hundred bases. So what you can get here is you can get one sequence of DNA, like this. And what we can get is we can get like let's imagine this was a 500 base pair, which is not uncommon. And we have, okay, hey, we have like 150 reads going out this way, and we have 150 reads going out this way, and then we have some other stuff in here. We know this wasn't usable, but we know there's how many base pairs are there. We can get a good idea of how many base pairs, even though we can't actually know what to call them. And the same thing going this way. So we have this of usable data, we have this of usable data. And the other thing is we actually know the entire transcript length, even though we can't call every single base in the middle, we know the entire transcript length. So what we can do now is we can map a lot of these, because I know I have whatever AT, 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 and then it goes on for a while, and then I know my sequence here as well, so I can anchor that somewhere else. And anchor that to other transcripts then that we have read. So what we can actually do is this improves our ability to align transcripts to each other, because even if we don't have usable reads all the way through, we still have a read, and we have a read, and we know how far apart they are. We have a good idea of how far apart they are. And so it helps us set how long that is.
Okay.
Okay.
Thanks.
No problem. That's good. Most people, like, when they don't know, like they totally miss it, they just kind of sit there. Okay, so pros and cons of RNA-Seq. What is going on with the other stuff before? First off, becoming the most used transcriptomics approach. And the other nice thing, unlike microarrays, this is going to give you absolute quantification of the number of transcripts. Remember with microarrays, because we're just getting fluorescence, this gives you relative quantification. And if you have some good things that you have a good idea that you can kind of calibrate your fluorescence to, you can get a really close idea, but it's not giving you the absolute quantification like you have in RNA-Seq. Now, the other thing that is really nice is that you don't absolutely need a well-annotated genome for this. So if you're using a non-model organism that there's no genome for, you can actually pull this off. You can't do that with microarrays. With microarrays, you need to have a transcriptome, you need to have the express sequence tags, you need to have a well-referenced genome, you need to have some idea of what transcripts you're going out and looking for, or else you need to have something to have those oligos to hybridize to on your chip. So this, you don't need that. You can go into a completely novel organism that you've never seen before in your life, do this process, and still get good information about the transcriptome. You cannot do that with microarrays. And that actually might be one good reason that SICB is using so much RNA-Seq, is most of the things that we're using there are non-model organism animals. I just thought about that, yeah. So that's probably why it's so skewed at SICB towards RNA-Seq and not microarrays, because so few people are working on model organisms like C. elegans or Drosophila or people or Dictyostelium. Yeah. So, and you can get whole genome coverage this way as well. So unlike microarrays, again, you are using, you're just looking for certain things necessary. And you can get, at this point for humans and stuff, you can get, I think, whole genome microarrays, but you don't always necessarily. Now, the cons of this, the downsides of this, is that reconstruction can be computationally expensive. To process this data, it takes way more computing power than to process microarrays. So you're going to be sitting down, and this is worse than phylogenetic reconstruction in terms of computational time. That being said, it's not, computers are getting better all the time, and this is way easier. Still takes the same amount of computing power, but there's so much more computing power in terms of desktops now than there was 10 years ago when RNA-Seq was just first being used. That it's getting relatively easy. And then also, it's still not, again, prices coming down all the time with RNA-Seq, but it's still more expensive than microarrays to actually do. So if you have a model organism and you don't have much money, a DNA microarray is still probably the best way to go. Okay, so let's talk about, just take a few minutes here now and talk about, in theory, what is now your process of actually analyzing RNA-Seq data. This is kind of giving you now a primer into lab where we will do some processing of RNA-Seq data and actually looking at some things with that. So the first thing that you need to do is that you have this FASTQ file. You have something that looks like this. And this is your raw output. What do we do with that? And the very first thing that you need to do with this, well, actually the very first thing that I'm kind of leaving out here is quality control. You actually go through here and high grade what reads are good and what reads aren't based on the quality information that you have there. And that is a very important step, but for time reasons, I'm going to kind of just gloss over that. Yes, you need to do quality control on your reads, like what we did for our Sanger data in lab a few weeks ago. But then once you do that, you need to then assemble this back into the transcriptome. Again, we just have these little chunks of DNA, so we actually have to go back and assemble this back into an entire thing. And there's a couple different ways that we can do this. Number one, we can do this with a reference genome. And we can have either a GFF file with that. So generally the reference genome is just going to be in a standard FASTA format. So we have this big old long, like really huge FASTA format for an entire genome, which is really big. And then you can either have with that either what's called a GFF file, which is a general feature format. You can either have that or not. The general feature format is what stores all the annotations for the genome. Have I talked about specifically what it means to have a genome annotated? So you have all of these sequences. You have this whole long sequence of a genome. That's great. You have all these A, T, C's, and G's. What does that mean? And actually, the process of going through and assigning gene names and functionality and going through and essentially figuring out what is the functionality or the parts of all this genome, that's annotation. So you have the sequence, but then you also need to know, okay, this stretch of DNA right here, this is a superoxide dismutase chain, as this particular one, or something like this. So you need to, the genome is kind of like, yeah, that's great, but not super helpful if you don't necessarily have annotations with it that actually say this stretch of DNA is this gene, or it is this regulatory element, or it is this histone interacting section, or something else like that. And so you can have the reference genome with the annotations, which is the most helpful. This is the easiest way to go about doing this, because then you just line your sequence of transcripts up with the genome, and then you can read back what is the function or the gene that this goes with. Now, it's not quite as simple as that, because the genome still has exons and introns together, and your transcripts will have those introns taken out, and you just are seeing your exons. And so you need to be able to open up gaps in a unique way without penalty. So the alignment that you're doing is somewhat different of an algorithm than the alignment that we were doing when we're just pairwise aligning our different sequences for like phylogenetic analysis or distance analysis between different sections, because opening up big gaps is a big penalty and it generally does not want to do that. In this case, you expect to open up these big gaps where you have these intronic regions. The other thing that we can have is a reference genome without the annotations, and these are kind of common as well. Now this might not be as helpful, but it does cut down the computing cost. Well, it might not be as helpful because you don't necessarily get back for each transcript what the gene necessarily is, but it does help with alignment immensely. Even if you don't have the annotation, this still is going to cut down your alignment time many orders of magnitude, because we can just find where it is in the genome, and instead of having to get overlapping reads to reconstruct transcripts, we can just use this huge genome that's already been quality controlled, hopefully, and find where it goes with that. Now, that being said, if you go this route, you need to actually go after this and find some way to figure out functionality for all these transcripts, and there's various ways of doing that. The other thing that you can use is you can use a transcriptome. So oftentimes for organisms that do not have a specific genome released, you oftentimes have a transcriptome. Someone else has already done RNA-Seq. They have already assembled de novo the transcriptome, or all the transcripts, and so that information, they make that information available because oftentimes you have to when you publish. And so that transcriptome data is available. If you have someone's assembled transcriptome already in place, it makes this incredibly easier as well. Again, you have completely assembled transcripts, and now you can just line yourself with where they go in that transcriptome, and life is a lot easier. And if you have none of these at your disposal, again, you can just de novo align these yourself. Going through, this is by far the most difficult, the hardest, takes the most computer power, but you can still get that to happen. We can still align transcripts again with their, or reads of transcripts with their overlaps, figure out what's going on. Now, that being said, certain things mess this up, like alternate splicing and other things like this make this sometimes the accuracy of this drop, but that being said, it still gets you somewhere instead of nowhere. And then you have several processes that happen. Again, if you have a genome with the annotations, or a transcriptome would generally become annotated as well because they need to make the annotations to do anything with the transcriptome. You just go through a process called transcript identification, which is just, again, pulling what is the function, or this transcript here, what is that doing, what is its gene name, what is it encoding for. If you don't have the genome with the annotation, so this is either genome without the GFF, or if you are having a you're de novo assembling this as well, you have to go through a process called transcript discovery, and then annotation. Annotation actually assigning the functionality of the name or the gene name to it. And again, this is more complex. I'll probably talk about this somewhat in lab, but not belabor the point right now. So that is the next section on that. Okay. So transcript discovery happens with the genome without the GFF. I guess it's transcript assembly that's happening without that, because again, we have to assemble the transcripts. Then we go through and actually do the annotation. Okay, so we're going to go through and we're going to get an alignment of all of our reads into individual transcripts. And the output for this is that aligned file is what's called a SAM file. And it gives you a whole bunch of information. I'm not going to delve into the entire thing. Every single transcript then is assigned at least 11 points of data along with it after the alignment. However, sometimes can be more than that. There is specifications for more pieces of data that can go with it. But at least 11 different fields are required for every every read. And some of them, one of them is the start position in the reference genome, transcriptome, or your assembly for this given read. And then it goes on for however long that read is. And we also have what are known as BAM files, which are just SAM files, but they are converted to a binary format. It makes the size a little bit smaller, which is really good because these can get to be fairly big when you have hundreds of millions of reads, and we're adding on 11 pieces of information for each one of them. So we make them into a binary. Now again, if you remember when we were talking about different ways of storing information, this means that unlike the text format files that we've been looking at so far, this isn't just human readable like that. You have to actually use some sort of interpreter to open that up. This is what a SAM file looks like. So you have header information here, and again, like what we had on the FASTQ files, all the header information for the entire file now is stored in these, or started out with these @ symbols, and we have all this. They give you some things like, this is the program that actually did the alignment. And then down below here, what we have is the actual, this is each actual read that we have. What we have here is just the name of the read first, and then I think this one might be where the reference genome actually aligns to. And by aligns to, what we mean is that this position right here, this is the base in the reference genome or the reference transcriptome or the assembly that this first base aligns to. And then we have all of this. We have quality information about the alignment through here, and then some other information that I'm not going to dive into right at the moment. But again, we have quite a bit of data that's going with each transcript now that gives it its alignment inside the genome. Once we do that, once we have this alignment of all of these reads, kind of one of the things that you really want to know then is, well then, how many of each of these transcripts are there? Which is kind of an interesting question to ask. There's some statistical methods that you can go about trying to determine how many transcripts do we have, or how many copies of each transcript occurred in the original data, or the original transcriptome that you're looking at. And to do this, we have to do some amount of normalization. So remember again that we have all these reads of the transcript fragments. And so there's a couple different things that make the probability given a certain transcript, it's more likely that you will see a read from that transcript. Number one, the longer a transcript is, the more likely it is that you will find a read for that particular transcript. Does that make sense? If you have a really short transcript and it's 100 base pairs long, a really small protein. And then we have another transcript over here that is 1,000 base pairs long. Okay? If those occurred at the exact same frequency in the transcriptome, we would expect to get about 10 times as many reads for the 1,000 base pair long, and actually slightly more than that because it doesn't exactly work that way. There's more overlap. When you consider overlap, it's slightly more than 10, but you expect to get about 10 times as many reads of the 1,000 base pair transcript than the 100 base pair transcript if they were expressed equally, just because when we fragment the DNA, the 1,000 transcript, the 1,000 base pair transcript is going to make more fragments. Does that make sense? Okay. Also is going to, because again, we want to know exactly how much. We don't really, it's nice to know the absolute quantity in there and not just the relative quantity. It also means the more reads that we do is going to equal more transcript reads. So the more times that the more sequencing we do, the more transcript reads you will tend to get for any given transcript. And that should hopefully make intuitive sense. If I do more sequencing, I would expect to get more sequences of any sequence I sequence. That was really kind of weird to say this. But does it make sense? Okay. So what we have to do is we have to get some sort of units that try to account for this. And there's a couple different ones that are commonly used. RPKM, which is the reads per kilobase per million reads. So this is essentially the reads per million reads is essentially giving you, and this is a per million total reads that we took. So essentially what this is giving you right here, the reads per million reads, is just essentially kind of like a percentage of all of the transcripts we got were this particular transcript. And then the reads per kilobase is then the reads per kilobase of that transcript. So then now we're normalizing it to the length of that transcript. So then we have the reads for the length of the transcript, so normalized for the length of the transcript, and then normalized for the entire number of reads that we got for the entire transcriptome. And this is the unit that they use if we are using a single-ended data. If you're using paired-end data, it's then fragments per kilobase per million reads. Slightly changing our definition. We're actually looking at how many different fragments, because again, you're going to get 2 different reads for every fragment, because we have the paired ends. Does that make sense? There's also other ways to look at this. We could just look at transcripts per million. This involves a little bit more processing than actually goes into this. This is fairly raw, and there's actually some more biases that enter into this that will skew relative amounts of, or this is not, the RPKM, the FPKM are not perfect representations of expression levels. There's more normalization that needs to happen. I'm going to talk more about that later, but doing some of that more normalization can result in transcripts per million. So this is a more direct, this is how many transcripts of this particular gene we had per million transcripts. Kind of a little bit more direct. And then this, we also have a TMM value, which is a trimmed mean M value, which corrects for a few other biases as well, including base composition. So actually, the sequencing process can be biased, and one of the ways that can be biased is by base composition. You can actually get higher amplification and higher sequencing done for high GC content transcripts rather than low GC content transcripts. because the GC bonds are stronger than AT bonds are, and so you can get biasing in that. And so if we correct for that biasing, also there can be suppression of rare reads. Let me explain how this works real fast. So imagine that we had, imagine we're looking at absolute quantification. And imagine we have 3 different sequence bots out there. I don't know. I'm trying to make this like just abstract from Illumina for a moment. Imagine we had like these little robots out there, right? That could just go out and sequence something, right? That's gonna go out and they're just floating around. They're like little nanobots that can just like sit on like a primer on DNA and just run through and just sequencing. So we have 3 of these that are like cruising through a cell, like doing its, like quantifying its transcriptome. I hope this isn't too abstract. And we have, imagine this for the sake of argument, we have one transcript A, we have 1 copy of that. And transcript B, we have 1 copy of that. And that's the entire transcriptome. The likelihood of both of those being sequenced is equal, right? We're probably going to get both of them. Okay. Imagine now we have the exact same number of transcript A. We still have 1. But now we have 100 million of B. We only have 3 little sequencing things, right? What's the likelihood that we are going to find transcript A? It's a lot lower. There's a very good chance we're going to miss it, or miss it more frequently. However, that being said, we still have not gotten a lower amount, but oftentimes it's going to look like we have 0 of that. So what you can get is if reads are rare, just because of their chance of getting missed, and a few other biases that creep into it that are a little bit more different than that as well. You can get suppression of rare reads, so they will end up looking rarer than they actually are. So some corrections that need to go into that. Following this, once you have transcript counts, we have some good way of quantifying the transcript. We go into the next thing, which is you want to compare this to something else. We want to actually see, okay, what do these other things look like? And how do they compare to other organisms, other cells, other tissues, other things like that. And that analysis is called differential expression of genes, or DEG analysis. And I'm going to get into that on Friday and on Thursday, because my idea is that I will have you do some DEG analysis on some transcriptome data in lab. Okay. It's time for me to kick you out of the room. So enjoy your Wednesday. I will see you tomorrow at 2 p.m. And we will do fun things together. Howdy.
Talk to you sometime about my data set, because I have one of them, but not the other one. I feel like I found them both at the same time.
Okay.
Because we mentioned, remember how I was doing mosaic?
Mhm.
Okay. We mentioned that we might want to compare it to some non-coding
But I think it's unlocked.
stuff to see
Yeah.
if there's something driving selection.
Yeah, got it.
I don't know what non-coding thing actually.
So one thing, you could just get some ribosomal.
Do that, so search for ribosomal.
Yeah, like ribosomal RNA. Yeah, like a 12S, something like that, or 16S.
Okay.
You know, something like that. Because even if it's under, that's under very, even if it's just reminding everyone just to. I mean, it's not that it's not.
I keep spacing. I need to
Yeah,
just
under very different.
I would like to meet Thursday.
Yeah, just so you know, I can meet either of those days.
No one responded back to my email, so.
So let me see here. Did you actually want to sit down and meet?
I don't know. Do you want to?
Yeah, I mean,
those days works for me.
Okay, if I have, I'll look for stuff, and if I have any questions, I'll just.
Okay, sounds good.
Because we don't really, yeah, okay. So then I'll just try and find some for the same species that I found. Or,
Yeah.
then when I run it, I'll just run 2 different cheeses and just look at differences.
I mean, it shouldn't
Yeah, I
take
think
long.
for this, for purposes of this, that would be good.
Okay.
Yeah.
And just try to do the same alignment, same types of
Yeah,
everything.
yeah.
Sounds good.
Yeah.
talk about
Cool.
where our classes are sitting.
Okay.
Tyler, how you doing?
Great. Well, I'm still waking up, but then that
Okay.
Did you receive the email I sent out yesterday?
I did.
Which had my attached file
And I was
the
working
date and
hard on
time.
this that I didn't, okay, so Friday, 9 to 10?
Yes.
Yeah, okay.
I just wanted to make sure everything went through because I was having some computer issues yesterday. So,
Okay.
Awesome.
No, yeah, I got it.
Thank you.
No problem. So now that should be in my schedule. Let me look if it is. And not yet. Wait, that's this Friday, not next. Wait. Oh, never mind. It's in the email.
Oh, he's back.
Yes.
That's next Friday or this Friday?
Next round.
Okay. I'll go
A
look at
week.
it next Friday.
From now.
Yeah, there it is. Look at that. Woo.
Yep.
Really? Okay.
Give you guys some time to read it.
Yeah, that's good. I'm harsher when I have just short times to read it.
Okay. And hopefully I've given you more than a week, so that's good.
It should be good.
Thank you, Dr. Allen.
No problem. Have a good day. How's it going, Matt?
Good, how are you?
Doing tired.
Getting tired?
Yeah, this would be nice to go get some caffeinated
juice.
beverage.